Hello everyone, my name is Jialin Hu. For the webinar of the course FWF 525, my topic is about the unconstrained ordination analysis. At first, I want to roughly introduce the ordination. Ordination is a statistical technique in which data from a large number of sites or populations are represented as points in a 2 or 3-dimensional coordinate frame, including constrained ordination and unconstrained ordination. Both of them are commonly used in ecology to visualize the multivariate data. The unconstrained ordination can be commonly used to visualize the mean trends between different sites in terms of their species composition or relative abundance. The constrained ordination explicitly includes 2 or more different sites of ecological information into a single analysis and directly examines relationships between sites of variables. That is, except for species composition, it also includes the matrix of environmental factors. But in my webinar, I will focus on the comparison of the unconstrained ordination methods. There are 3 very commonly used unconstrained ordination methods. The first is non-metric multidimensional scaling. In short, we call it NMDS. The second is principal coordinates analysis. And the third is principal component analysis. The multidimensional scaling is a means of visualizing the level of similarity of individual cases of a dataset. It is an unconstrained method, using only the rank order of intercase dissimilarity values. When we conduct non-metric multidimensional scaling analysis, a small number of axes are explicitly chosen prior to the analysis, and the data are fitted to those dimensions. There are no hidden axes of variation. It is based on a numerical technique that iteratively seeks a solution and stops computation when an acceptable solution has been found. The principal coordinate analysis is also a method to explore and to visualize similarities or dissimilarities of data. It is one of the classical methods of multidimensional scaling. It starts with a similarity matrix or dissimilarity matrix and assigns for each item a location in a low-dimensional space. But it is a kind of eigenanalysis and calculates a series of eigenvalues and eigenvectors and tries to find the main axis through a matrix. The principal component analysis is a statistical procedure that uses an orthogonal transformation to convert a set of observations of possibly correlated variables into a set of values of linearly uncorrelated variables called principal components. It is the simplest of the true eigenvector-based multivariate analysis. In the result of the principal component analysis, the first principal component accounts for as much of the variability in the data as possible, and each succeeding component accounts for as much of the remaining variability as possible. For the case study, I provided 131 16S rRNA sequencing datasets about different air environments and activities large environments to see the dissimilarity of airborne bacterial communities between air and the bacterial communities of activities large in the same wastewater treatment plant, and the difference of bacterial communities among different air environments. The air environments including air collected from wastewater treatment plant, suburban, traffic lines, smoke and haze, hospital, urban, atmospheric biofilm, and swing building. The 16S rRNA sequencing data could be used to reveal the bacterial communities of the environmental samples. I will use R to visualize the dissimilarity of the microbial communities among these environmental types, and the data should be placed into a big matrix like this. The rows represent the different samples and the ranks represent bacterial genera. The data are the relative abundance of the bacteria genera. This is the R command for non-metric multidimensional scaling. When we input the commands into R, and we will see the 20 runs, and the stress value is the non-negative number that reflects how well the ordination summarizes the observed distance among the samples. The nearer the stress value approximates to zero, the higher confidence the result has. The value of stress less than 0.3 means that the result has high confidence. So from our result, we could say that all our stress value below 0.3, which suggested that our result had a very high confidence. This is the non-metric multidimensional result based on Baker distance of the distribution patterns of bacterial communities in our 131 different environmental samples. The left figure is the initial result of the from the R, and the right figure is the result after beautification by Microsoft Visio. In this figure, each point represents one sample, and the different colors mean different environmental types. The closer the points, the more similar bacterial communities the points represented samples. For example, the wastewater treatment plant air samples are represented as light blue points. The activities large samples are brown points, and the other 11 air environmental samples are environmental types are represented in other colors. The results indicated that the airborne bacterial communities of wastewater treatment plant showed a significant distinct distribution patterns from the bacterial communities of other air environmental types, even distinct from the suburban air collected in the same city, just like the three blue points, but it was comparatively more similar to the bacterial communities of wastewater treatment plant activated sludge. It was represented as the brown points. This result suggested that the activity sludge may affect the airborne bacterial community in wastewater treatment plant. Besides, the most investigated city air environmental samples were clustered distinctly but with some overlap, which verified that the samples from the similar environmental types mostly cluster together independently of their geographical locations, because the most of the city air samples were collected from different cities, but they cluster together. And this is the argument for principal coordinate analysis. This is the result of principal coordinate analysis also based on Bray-Curtis distance. We could find that the result from the principal coordinate analysis looks the same with the non-metric multidimensional scaling results. I think the reason may be that the principal coordinate analysis is also one of the classical methods of multidimensional scaling. So they showed the similar result. This is the R command for principal component analysis. From this result, we could see that the principal component analysis is not appropriate for the analysis of the dissimilarity of bacterial communities of our datasets, because from the figure, most samples are clustered together, and it can't well reflect the dissimilarity of the bacterial communities among different environmental samples. In summary, for the analysis of 16S RNA sequencing data to compare the dissimilarity or similarity of bacterial communities among different environmental types, the non-metric multidimensional scaling and principal coordinate analysis are commonly used, but the principal component analysis may not adapt. But compared to non-metric multidimensional scaling, most ordination methods In most ordination methods, many axes are calculated, but only a few are viewed owing to graphical limitations. But the non-metric multidimensional scaling, a small number of axes are explicitly chosen prior to the analysis, and the data are fitted to those dimensions, so there are no hidden axes of variation. Besides, most other ordination methods are analytical and therefore result in a single unique solution to a set of data, but the multidimensional scaling is a numerical technique that iteratively seeks a solution and stops computation when acceptable solution has been found. In other words, it stops after some pre-specified number of times. So the non-metric multidimensional scaling ordination is not a unique solution, and a subsequent multidimensional scaling analysis on the same set of data and following the same methodology will likely result in a somewhat different ordination. Finally, the non-metric multidimensional scaling is not an eigenvector technique like the PCA or PCOA analysis or correspondence analysis that ordinates the data such that axis 1 explains the greatest amount of variance, axis 2 explains the next greatest amount of variance, and so on. So the non-metric multi-dimensional scaling ordination can be rotated, inverted, or centered to any desired configuration. It just shows the relative location of different samples. That means in the figure from non-metric multi-dimensional scaling, the distance between 2 points just show the dissimilarity or similarity extent of the two samples, not considered the axis explanation.